Hello, and thank you so much for the invitation to speak today. We're really excited to talk about some of our latest developments, some of which are very late-breaking at Optigo. Basically, our goal is to do what many others are trying to do, and we think we have a very unique embodiment of our goal, which is to reduce the frequency of intravitreal injections for all sorts of disease states. And as you all know, retinal injections are on the rise and are skyrocketing almost exponentially. However, that treatment burden has led, as all the publications have shown, to worse efficacy in the real world than we see in the phase 3 trials because of access to drug, cost, etc. We also know from the market that durability defines success, and every time a drug comes out that has better durability, its sales surpass those of its predecessor. We see Lucentis, Eylea, Vabysmo here, but when we look at our drug, OPT-640, which I'm going to talk about today, our durability is going to last at least 180 days, hopefully a year or even longer, no monthly doses required for loading doses, and reduce the injection burden by at least 70% or more compared to Vabysmo, which would get us to a very high projected sales rate. Lots of other long-acting delivery approaches have failed to achieve their promise, and this is devices, sustained-release polymers, new biologic entities, gene therapies, and other classes have fallen short, really, of their promise. How are we trying to achieve our aim? Our target product profile, again, is a 6 to 12-month injection frequency, maintaining the efficacy of existing standard of care treatments while extending the duration. And this is how we're doing it, and this is our secret sauce. We are not intending to put a polymer or implant or insert into the eye. We are simply taking the hydrogel polymer that Mother Nature put in the eye, which is called the vitreous, and we all know it well, and using that as our sustained-release polymer by binding existing drugs to it non-covalently in a way that, like a Post-it note, the drug can bind and fall off. Very simple. And by titrating the binding affinity between whatever the drug is, and our first embodiment is using aflibercept, the parent molecule of Eylea, and whatever the target is in the vitreous, and there are 3 that we went after, I'll show you in a moment, by titrating that binding affinity, we can control exactly what % of drug at any given point is bound versus unbound and ready to go and gobble up VEGF or whatever its target is. And this is what we've really done. We've put in an enormous amount of effort, years and years, and a lot of money in terms of developing libraries of binders and then aflibercept-based molecules based on those libraries that bind to collagen type 2, hyaluronic acid, and albumin. And so we actually created thousands of molecules, screened them in vitro, moved the best ones to in vivo studies, and then from that moved on to even later stage in vivo PK and efficacy models in rabbits, and then later in NHPs, all to come out to our lead candidate, which is OPT-640, that I'm going to focus on today. And this is OPT-640. It's aflibercept that is targeted to the hyaluronic acid, or HA, that is in the vitreous of every person's eye. This is a bifunctional fusion protein, to use a term that Emmett just stated earlier. It's not 2 different molecules coupled together. It is expressed as a single protein in mammalian cells without any chemical coupling involved after the fact. As you can see, this HA binder, which is in the bottom in red, is actually not involved in the business end of the aflibercept molecule. So the efficacy is not impacted. And what's great about this is that our HA binder, as well as all of our other binders, can be used as a platform with any other intravitreal product that's available today or in the future. And so what do we see? We did rabbit studies first, as I mentioned earlier, increasing the half-life compared to regular aflibercept 5-fold with an increase in the area under the curve, or AUC, 8-fold. And we see this increased concentration across different components of the eye: aqueous humor, vitreous humor, retina, RPE, choroid. And we then went on to additional studies. We did a rabbit VEGF challenge model to see, do we actually carry this PK advantage into an efficacy advantage? And yes, we increased the duration of activity of aflibercept by 3.5 times or more with that very same molecule, OPT-640. And was tolerability good? The answer is yes, in terms of ocular exams as well as histopathology. clean profile. So then, most importantly, here's our late-breaking data. We've done a very exhaustive cynomolgus monkey PK and tox study, and we really managed to blow our TPP out of the water. What did we show? Looking on the left-hand panel with a single injection, you can see in blue is our product, in gray is what aflibercept would be, and at day 42 post-injection, we see 100 to 1,000-fold more drug in the eye, which is incredible. 8 times half-life extension versus aflibercept, which allows us to do 8 to 12-month or even better dosing. No monthly dosing, loading doses are required with this. In fact, if you do monthly loading doses, as we showed in the panel to the right, you get way too much drug in the eye. We know that there's free drug available because we see a lot of it in the aqueous humor, so it's not just all bound up to vitreous. It's a question that we frequently get. And so we're seeing 24 or 0.8 day half-life versus a 3.1 day half-life for aflibercept. Incredible. The tolerability is comparable to what we see with other anti-VEGFs in NHP models. So it's looking incredibly good in terms of tolerability as well, and this is what we're now intending to advance into the clinic. This shows you our development roadmap. We've really thought all the way through approval here. We're currently at a stage where we're raising a Series A round of approximately $50 million to take us through IND and our first-in-human trial. Our intent is to do a very robust first-in-human that is not just an open-label phase 1, but a robust, compared to aflibercept, phase 2 trial that will make us phase 3 ready with the intent to go into phase 3 within 3 years or less. Our leadership team is very experienced. We have on our board as an advisor Udo Vetter, as well as members of the Vetter management group. These are folks who are heavily involved in the development of manufacturing for anti-VEGF drugs that are currently on the market. We have an amazing group of KOLs and advisors, including David Boyer, Bob Avery, Arshad Khanani, and so many others who are here and not here. Just to finish, well tolerated across multiple different species, prolonged half-life in multiple different species, prolonged efficacy, again in multiple species, and we have NHP study that's now wrapping up with a major opportunity ahead of us. So thank you very much.